Hello students, today I am here with first part of Animal Kingdom of 11th standard. Here we will discuss about basis of classification of Animal Kingdom, level of organization, symmetry, Diploblastic and Triploblastic, Siloam, segmentation and notochord. So let us start the session. First of all, we will study about what are different bases on which the animal kingdom is classified. Animals are classified on the basis of arrangement of cells. Animals are multicellular, heterotrophic organisms. In few organisms, the cells are loosely arranged, whereas in others are organized into functional units of progressively increasing complexity. Another basis is body symmetry. Symmetry refers to the similarity in the arrangement of parts of opposite sides of the body. Symmetry may be asymmetry, radial symmetry or bilateral symmetry. Later on, we will discuss in detail about all this. Animals are also classified on the basis of nature of Siloam. Siloam means body cavity. Certain animals have no body cavity except that in the digestive tracts, and other possesses an additional body filled cavity between the body wall and digestive tracts, and some have pseudocoelom. So this also we are going to discuss here. Next basis is patterns of digestive system. It may be incomplete digestive system or complete digestive system. Incomplete means there is only one opening for intake of food as well as elimination of feces. But in complete digestive tract or digestive system, they have two different external openings, that is the mouth for intake of food and cloacal aperture or anus for elimination of feces. Another basis is circulatory system. It may be of closed type or open type. Animals are also classified on the basis of reproduction and developmental processes. Fertilization may be external or internal. Cross or self-fertilization. They may be oviparous or viviparous. Further development may be direct or indirect. Thus, we have seen that here there are different bases for classifying animals. Now, let us discuss about level of organization. Animal body show different level of structural organization. It may be cellular level, tissue level, organ level or organ system level of organization. Cellular level is found in sponges. In this case, body consists of many cells, but the cells do not form tissues and the cells are loosely arranged. The cells show some division of labors but cooperate little with one another and are mostly independent. In coelenterates, the body is multicellular and the cells occur in two distinct layers or tissues of specialized cells. In some organisms like Platyhelminthes and other phyla, it exhibits organ level of organization, meaning thereby is that tissues are organized in the form of different organs to perform separate functions. Here you can observe the flow chart of level of organization. It starts with non-living atoms, then molecules, further from non-living, living organelles, cells, tissues, organ system and organisms come. Animals belonging to annelids, arthropods, mollusca and echinodermata as well as chordates show organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexity. For example, the digestive system, it may be complete or incomplete as we have discussed earlier. Complete digestive system means 2 openings, mouth and anus, whereas incomplete means 1 opening for both intaking of food as well as removal of feces. As far as circulatory system in animal is concerned, it is of 2 types. They may be open type or closed type. So let us see what are the differences between these 2 types of circulatory system. In open type, the blood is pumped out of the heart and the cells and tissues are directly bathed in it. It is commonly seen in arthropods and mollusca. In closed type, blood is circulated through a series of vessels of different diameter through arteries, veins and capillaries. Annelids and chordates have closed circulatory system. This figure shows cellular level of organization. Here you may observe different cells like epithelial, connective, muscular and nervous cell or nerve cell. Now let us discuss about symmetry. As we have discussed earlier that symmetry means the similarity in the arrangement of parts on the opposite side of the body. Some animals like sponges, they are highly irregular in form that their body cannot be divided into 2 equal parts by any plane that passes through the center. Such animals are said to be asymmetrical. Certain animals such as member of coelenterates, ctenophora and echinodermata have a body with a number of similar parts radiating out from a central axis. So the body can be divided into 2 equal halves by any plane passing through the center from top to bottom. This type of symmetry is called radial symmetry. In all vertebrates and most invertebrates, the chief organs of the body are paired and are arranged on the sides of a central axis connecting the head with the tail. Thus, the body can be divided into identical left and right halves in only one plane and it is called bilateral symmetry. For example, it is seen in annelids, arthropods or chordates. You can see here the butterfly and it is the bilateral symmetry. In this figure you may observe the asymmetrical, radial and bilateral symmetry of different organisms. Now it is about diploblastic and triploblastic organisms. The early embryo acquires fundamental cell layers and these layers are called as germ layers. When cells are arranged in 2 embryonic layers, an external ectoderm and internal endoderm, then it is called diploblastic, for example in coelenterates. In such cases and an undifferentiated layer, that is mesoglia, is present in between ectoderm and endoderm. But in those animals in which the developing embryo has a third layer of mesoderm, that is in between ectoderm and endoderm, then it is called as triploblastic. It is seen in platyhelminthes to chordates. Here you may observe the diploblastic and triploblastic organization. You see that in diploblastic it is of 2 germ layer, endoderm and ectoderm, and in between ectoderm and endoderm mesoglia is present. Whereas in case of triploblastic organization there are separate 3 layers: ectoderm, endoderm and mesoderm. Now our next topic is Siloam. Siloam means body cavity. It is of three types: coelomates, pseudocoelomates and acoelomates. When the body cavity is lined by mesoderm, then it is called as coelom and the animals are called as coelomates. It is present in annelids, mollusca, arthropods, echinoderms, hemichordata and chordata. But in some animals, the body cavity is not lined by mesoderm, but the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such body cavity is pseudocoelom and the animals are called as pseudocoelomates. For example, it is seen in Aschelanthes. The animals in which the body cavity is absent are called acoelomate. For example, in Platyhelminthes. This flow chart and diagram represents about the acoelomates, pseudocoelomates and coelomates. In case of acoelomates, animals is without coelom. Example is Platyhelminthes. In pseudocoelomate, body cavity not lined by mesoderm. Mesoderm present in scattered pouches and this is present in Aschelanthes. And the last coelomates, here animals are having true body cavity, that is coelom, and examples are annelids, arthropods and chordates. Now the last topic of today's session is about segmentation and notochord. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. This is called metameric segmentation. What it is called? It is called metameric segmentation and the phenomena is called as metamerism. It is commonly seen in earthworm. Now it is about notochord. Notochord is the rod-like structure. It is derived from mesoderm. Notochord is formed on the dorsal side during embryonic development in some animals. Animals with notochords are called chordates and those animals which do not possess this structure, that is notochord, they are called as non-chordates. So porifera to echinodermata are non-chordates. This is the outline classification of animal kingdom based on common fundamental features. You can see here it is divided, the level of organization is cellular, tissue, organ system. Further, the radial symmetry that is present in coelentrates, ctenophora, bilateral symmetry that is present in Platyhelminthes, Aschelanthes, annelids, arthropoda, mollusca, echinodermata, hemichordata and chordata. So children, this is all about today's session. Thank you. Have a nice day. For any kind of doubts, please write your question in comment section.